Well, I really appreciate the opportunity to be here again in London and amongst friends and colleagues to open this very exciting meeting. I first will do a bit of reflection on the past in ME/CFS, and then hopefully give you some inspiration in the introduction of what you're going to see today from recognized world experts. With respect to the theme of infection immunity and mainstreaming ME research. So I have my biases. You know, the federal diagnostic criteria were established 25 years ago, and they've been revised many times and debated continuously, and we still debate. And I really make a plug for the need for a single set of diagnostic criteria that can be accepted worldwide by researchers and clinicians for CFS-ME, or else well-characterized subsets, and forget the nomenclature. Forget chronic fatigue or ME, and look at the pathogenesis of subsets. So reflecting on the history, you all know that in the pre-80s, there were outbreaks of a disease largely characterized here in the UK that can now be described as CFS-ME. Then in the 80s and 90s, when I entered this field, the focus was really on the fatigue, and hence the definitions that were established by the CDC and other agencies. By the mid-90s, we were moving on to focus on the neurological, immunological, and endocrine post-exertional malaise. Most of the experts in this field now think those aspects are critical to the diagnosis of the disease. This led to some new definitions as well. These criteria have been around for 25 years, and I personally think it's time to stop counting, querying patients, and to begin the development of real intervention in diagnostics. So you know the prevalence in the US, about 1 million people. In the UK, 1 to 2% of the population. Worldwide, 19 million. That's not a rare disease by any standards. It's not an orphan disease. It's very significant. What is known worldwide about CFS-ME is that it's similar amongst all patients. With respect to symptoms, presentations, physical exam, there's a high degree of activity limitation. I don't have to tell anybody in this room that. They do experience great socioeconomic disadvantage in many ways. And lastly, there's pure and simply insufficient care, unmet medical needs, and unmet home needs. It's costly to treat. The estimated cost to the United States is $9 billion in direct costs, $51 billion in indirect costs if you consider loss of work, etc. So what's the dilemma? Well, I think the diversity of presentation and the pathogenesis really confounds clinicians and researchers alike, which sometimes brings us into a paralyzed state. There's a model for the pathogenesis of chronic fatigue syndrome that I think most of the experts would generally agree with, and that's that there's a genetic predisposition, perhaps gene expression modification. Then there's an event or triggering event, whether an infection, trauma, stress, or immunizations. Then a mediation with the immune system, neuroendocrine, etc., that results in the symptoms of ME/CFS. And you'll hear from a number of speakers today who have special interests in each of these phases with respect to their research and current, even some diagnostic and therapeutic suggestions. So traditional medical diagnosis had 4 steps that we physicians are all very familiar with, and we do 30 times a day. That's achieve the most specific diagnosis, determine the severity and prognosis, institute therapy, and then assess the efficacy, safety, and quality of life of the therapy. These concepts are even used by institutions such as the FDA and biopharma to assess new drugs, interventions, and diagnostics. Now, the challenge is that that traditional model doesn't fit so well for chronic fatigue syndrome ME, and these are the reasons. It's a distinctly heterogeneous population that confounds the diagnosis. The CDC's recent study at 7 sites has documented that even the experts don't see the disease the same and define it the same way. There's no clear-cut biological marker that's been validated and accepted. There are multiple clinical definitions. There's no licensed drug, even after 25 years, and there's no universal surrogate marker. So symptomatic therapy, I think, may alleviate symptoms and improve the quality of life, but in my experience has never returned a patient to full cognitive and physical functioning. So as Dr. Leonard Jason said in a recent scholarly review, subsets of patients have to be defined that have evidence of a pathogenetic mechanism that can be treated directly. You may know that the FDA recently had a stakeholders meeting, the first that they've ever done with respect to chronic fatigue ME, in which they solicited the input of physicians and patients with respect to drug approval for chronic fatigue syndrome. And I'll just give you some highlights from this meeting because it was, as Dr. Gibson has pointed out, a step in the right direction for us that's taken many years to accomplish. And I was struck by certain features. The first was that the FDA was totally unenlightened with respect to the nature of CFS, the disability that's attended, the lack of appropriate treatments, and the vast amount of research that's already been accomplished in the field. There were key take-home messages from this meeting. The first, that a biomarker is not necessary for a drug approval for a syndrome or condition that can be confirmed by some other or defined by some other mechanism. This is the bummer for patients. Drug development takes 8 to 15 years, and it's largely supported by big biopharma and patient-driven by market potential. And the problem in CFS-ME is large biopharma has not been interested, and the patients have been relatively impotent in terms of approaching the big pharma as well as the FDA. Now, drug repurposing can sometimes be utilized to shorten this process, and that's utilizing a drug that's already in the production line or licensed for a new indication. The FDA requires well-documented, validated, and reproducible endpoints, which we simply don't have in this disease, although you'll hear it later today about some suggestions with respect to that. Endpoints can be surrogate markers, but the problem with surrogate markers is they're very difficult to validate for a disease process. So along comes Invest in ME, my favorite small charity with big cause, with the goal of etiology, pathogenesis, and epidemiology of ME, to present some potential solutions today to these challenges of mainstreaming ME research. One is computational analysis and bioinformatics. We have a world expert on that topic. We're going to hear about novel pathogen discovery, immunological biomarkers, endpoint evaluation and study design, and even drug development and clinical trials. Pretty exciting day planned. Computational analysis is a new and exciting advancement in the field of medicine. Totally new to me, being of my generation. Large amounts of collected data, such as genomics, can be interpreted and subsets determined for autoimmune patients, persistent or chronic viral infections, immunological abnormalities, or even autonomic abnormalities. This is not only limited to screening biological samples, but I'll show you an example of this. CFIDS Association of America used computational analysis to model drug repurposing. This was a large project that they funded, and they determined two very interesting things. They came up with a list of drugs that should theoretically be useful in the treatment of chronic fatigue syndrome ME. But they also came up with the finding that most of the drugs we currently use actually worsen your symptoms and perhaps should not be utilized. And that was all by this technique of computational analysis. And although there's much that computational analysis can tell us, there's confounding factors in chronic fatigue ME. The samples have to be from the right source. Many studies have looked only at peripheral blood. We know that many pathological processes and even pathogens have specific tissue and are rarely detected in peripheral blood. Last year, I talked about the onset of the novel Pathogen Discovery project going on at the Center for Infection and Immunity in New York City. It was conducted in 2012. The final analysis is underway. There's a large sample repository developed, and the biological samples including saliva, feces, tears, plasma, serum, and even urine. A separate study that's going on underway is looking at the spinal fluid from 60 CFS-ME patients and 60 MS controls. And hopefully we'll have some information with respect to those studies from one of the later speakers. Immunological biomarkers, I think, are critical to diagnose and establish the prognosis. These can define subsets of patients that are amenable to treatment, such as cytokine blockers or immune regulators, such as Ampligen. I myself have a personal favorite, NK cell function and enumeration, and I think it has great potential as both a diagnostic marker and objective endpoints in clinical trials. There's a large literature with respect to NK cells in subsets, cell function, phenotype, microRNAs, and even NK cell genes. And it's a field that there's quite a degree of agreement with around the world with respect to utility. So the FDA spent quite a bit of discussion on endpoint evaluation. As you all know, in January, the FDA denied the approval of Ampligen, largely because of a lack of objective endpoints that determine drug efficacy. Again, these endpoints that were suggested that could be used included NK cell function or VO2 max and a proper large conducted trial. An example that was used was that the PACE trial here in the UK used 6-minute walking test pre and post intervention to determine an endpoint improvement. This is a test that's refuted by experts. It's very subject to effort and is not considered an objective endpoint of functional capacity. A VO2 max test, however, is universally accepted and endorsed by the American College of Physicians in order to determine disability status and physical function. So study design was the last point that the FDA was very concerned about, and that they made a very clear point that large number of patients are needed in order to conduct useful and meaningful studies. And I think many of us have erred in this, probably because of budgetary restraints in doing very small, isolated studies. Smaller studies can be meaningful only if they have a highly homogeneous and similarly defined population. Most smaller, non-blinded or non-placebo-controlled intervention studies, I think, will not be very useful. Drug trials are needed in this field, but they must be large, use accepted endpoints, and have well-characterized patient populations. We'll hear later today about a drug that's already approved. So it's very far along this drug development pipeline. It's right at the end. It's actually in the category of being repurposed. And we'll hear about rituximab and the studies from the Norwegians, and I'm anxious to hear an update. But I'll make a point that we really need a large multi-site clinical study with the characteristics of universally accepted and validated endpoints, a very specifically defined population, and then, of course, perhaps one of the biggest obstacles is appropriate level of funding. So in summary, on reflecting on this field over a number of years, I think worldwide collaboration is necessary to define the subsets of patients based on universally accepted biomarkers, to determine more specific diagnosis and prognosis, to define the exact pathogenesis and mechanism of disease, to design appropriate interventional studies that would meet regulatory criteria for licensing, and lastly, to create CFS-ME centers of excellence where patients worldwide can actually receive the best and newest treatments. I thank you for your attention, and I look forward to the day with all of you.